Well, it's a pleasure to be here. I appreciate you all coming to a conference on data and then attending a talk on medicine. So before I get going, I always have to start with a disclaimer. So I'm actually just like all of you. I am a computer scientist, and now I do medicine. So if there's anything in this talk that looks like medical advice, for God's sake, do not take it. My highest formal qualification in medicine is, in fact, a C in high school biology. But this is, in fact, what I do full-time now. So for most of my academic career, I actually did stuff like cybersecurity, high-performance computing, but actually mostly just programming languages. My PhD is actually in static analysis of higher-order programming languages and has no bearing on what I do these days. But I also want to point out that this fusion of computation and biology is by no means new. In fact, if you look at Alan Turing, you will find that he published a paper in biology. In fact, it's on the morphogenesis, or how you go from an embryo to a complex shape like you and me. In fact, if you look at Google Scholar for Alan Turing, you will find that it is his most cited paper. So Alan Turing, the father of computer science, is in fact more biologist than computer scientist for his use of reaction diffusion systems to describe that complex biological process. And I think it's in part of the fact of the way that Alan Turing thought that led him to this major insight within biology. So I'll make a claim today, and that claim is that data is actually the greatest drug of the 21st century. And I will argue that precision medicine is the vehicle by which we will deliver data as a drug. So if we're going to deliver data as a drug, then obviously we need an algorithm for precision medicine. And by the end of the talk today, I'll have sketched the outline for doing exactly that, along with some examples of how we do it in practice. So what exactly is precision medicine? Well, I go back to the launch of the Precision Medicine Initiative under President Obama, and when he launched this program, he gave it a definition. He said it's really all about delivering the right drug to the right patient at the right time. And then as soon as he said that, there was this explosion of people afterwards trying to put new terms on top of what precision medicine really meant. And so I've spent much of the past couple of years actually trying to rein it back into a single unifying definition, and I think I've found it. To me, precision medicine is really all about optimizing health with data. And so if you're using data to optimize the health of an individual or of an entire population of patients, you're really doing precision medicine. Now, some physicians will disagree with this. They'll say, but I've always tried to find the best possible treatment for my patient. How can you possibly say I wasn't doing that? And I'll say what's changed is the amount of data you can collect on an individual patient, and therefore the amount of computation you have to do to find the optimal treatment for a patient. So for me, this process looks something like this. You start with a patient. Your goal is to find that best possible treatment. So you start by collecting their data. Now, the piece of data that got everybody really excited about precision medicine was the clinical availability of the human genome. The fact that you could look into our DNA at every single letter and actually have essentially the blueprint for what makes us, us. And that led to the promise of these ultra-individualized treatments. But I'll argue that all forms of data that you have on an individual, including things like electronic health record data, Fitbit or Apple Watch data, all of that data still counts in this complex optimization process. So I do think of precision medicine as solving this constrained optimization problem, where we're trying to modulate things like lifestyle and treatment to find the optimal health for any given patient. So this is oftentimes why it's also called personalized medicine, because it's all about finding the right treatment and the right lifestyle for any given patient to find their individual optimal health. I'll also tell you that in practice, we don't always, even with as much data as we can collect currently, we don't always have enough data to find that best possible treatment. So sometimes we end up recommending a scientific experiment on behalf of an individual patient in order to find the data that we need to find, or even pose that constrained optimization problem in the first place. So to see what this looks like in practice, what I'd like to do is just tell you sort of my personal journey through precision medicine that brought me to this stage today. And for me, it really begins with my son Bertrand. So he's 11 years old today, but this is him back when he was a year and a half old. So by the time he was 6 months, we knew something was wrong, and we landed in a place called Undiagnosed Island. This is if you've ever known somebody who's had a patient or a family member who's been undiagnosed for a long time, this image of being trapped on an island alone, not knowing what the problem is, will generally resonate with them. And when we were trapped there as a family, we didn't know why it was that Bertrand was suffering. We didn't know why he had seizures, sometimes hundreds of seizures per day. We didn't know why he had severe developmental delay. We didn't know why he had a movement disorder. And we didn't know why, for some reason, he could not cry tears. I mean, he could cry and go through the emotions of crying, but he would never make liquid tears. And so we were trapped there for 4 years. So I'll speed it up a little bit and describe the process and how it went. So after 4 years, Bertrand became one of the first human beings to ever have what's called his exome, which is an important subset of the genome sequence, and use this in a diagnostic capacity. And so just to short circuit what this process is like, when you're looking in someone's human genome for the cause of their disease, it's a pretty daunting process because the human genome, if you represent it as strings, it's 6.5 billion characters. It's big. And what I'm saying is we believe that any given genetic disease is typically caused by 1 or 2 mutations, really 1 or 2 typos in that giant string. And your goal is to find it. So to put this in perspective, imagine that you had 800 4K TVs, and there was 1 single broken pixel among them. That's what you're looking for in terms of rare genetic disorders. And yet somehow we're able to do this now. I'm not going to go into a lot of the details here, but we've developed a number of algorithms that allow us to pull out the most promising mutations in terms of what's potentially driving disease for this process. And it actually worked for Bertrand. So we figured out through a team of scientists at Duke University that he was, in fact, the very first human being ever to have this new disease called NGLI1 deficiency. So people had it before, but no one had ever observed this disease in the wild. It was essentially undocumented in the scientific literature. And what had happened was he's effectively missing the function in this gene called NGLY1 or NGLI1. So that happened when he was about 4 years old. And then I said, well, we got to do something to actually help him, because they will also tell you in an event like that there's literally nothing you can do. Because, hey, if someone's ever even seen the disease before, there are obviously no treatments for it. So a couple things happened. I jumped into the basic science around this disease. I'm not going to go into the biology of this gene. It doesn't really matter for the purposes of this talk, but I did a lot of basic science. I learned a lot of glycobiology, essentially. But the other thing I did was I said, there has to be a way to find a community for this disease, to bring other patients that have it together so we can all work on this disease, because you can't just have one patient versus an entire disease. So the other thing I did in tandem with the science was I wrote a blog post. And that blog post really had to do 2 things. It had to go viral, and it had to rank very highly in search results for the kinds of symptoms or search terms that other patients were likely to type in. So this is the post that I wrote. And the picture of Liam Neeson, the clickbait title, all that is really just to get people to click on this thing and pass it around and make it go viral. And it actually worked, because the internet is kind of sadly predictable like that. Now, honestly, I probably could have put a cat picture there, and it would have worked just as well. And there's 5,000 words beneath this designed to really help out with the search engine optimization so that other people would link to it and it would show up for all the right search terms. So it actually worked. In fact, a couple weeks ago, I was in Germany, and anytime I travel, I try to meet patients around the world for this disorder. So I met a patient for this disease a couple weeks ago in Germany, and today we found about 70 patients that have this disease. This is about 7 years later now, and continue to find more all the time. So what's amazing is that we estimate there's probably a few hundred patients around the world that actually have this disease, and somehow we've managed to stitch together a collective of a large fraction of all those patients. This also led to an article in the New Yorker, and all these things led to other things. The article in the New Yorker led to my involvement in something called the Undiagnosed Diseases Network, this big NIH-funded project. It also led to the NIH doing a study on this disease, where they actually enrolled all the living patients that we could find and began to bring them 1 week every year to NIH in Bethesda, Maryland, to actually study them. And this continued to snowball. So the advisory position at the UDN led to a faculty position at the Harvard Medical School. Again, despite the fact that I have a C in 6th grade biology, I'm somehow on the faculty there. And then, if you start to combine the insights from the natural history study, studying all these patients with the basic science we'd done, about 2 years after the discovery of this disease, I was finally able to predict a potential treatment. I predicted that these patients had a metabolic deficiency, meaning there was some metabolite that they were missing in their cells as a consequence of this missing gene. So it happens to be called N-acetylglucosamine. It's no secret, it's out there in the open now. But back then when I sort of stumbled across this and had this theory that he was missing this, I said, I don't know what to do. So what I did is I Googled it. I found out that you can buy this stuff on Amazon.com, so I did. It showed up on my doorstep 2 days later, thanks Amazon Prime. And literally just took a jar of it, I think 60 grams, and I just ate it all in one sitting. And I woke up the next morning alive. And that, for me, concluded FDA phase 1 safety testing. Because in my household, I am the FDA. Or I guess over here it's the EMA. But that's how it works. So then I ended up deciding to give it to Bertrand. And the amazing thing was, after about 3 days on this, in a disorder where no child had ever cried tears, he was actually making his first tears. So I didn't know what to expect when I started this, but that's what happened. And I didn't know if it would help with seizures. I didn't know it would help with the movement disorder. I didn't know if it would help at all, but tears is what it did. And this is significant because at this point in Bertrand's life, they were recommending what's called a tarsorrhaphy, where they were literally going to sew his eyes shut to protect his remaining vision. Because if you don't make tears, you have serious eye problems. He'd had surgery on his eyes to drain the pus from the infections that you would get when you don't make tears. And in fact, a lot of the kids that we find that are older have serious or severely damaged vision. So I did what any parent would do when they see their child crying for the first time. I actually did collect his tears, I packed them on dry ice, and I shipped them to a lab in California for analysis. And it's what most parents do, I guess. And in fact, those tears yielded an ocean of science about the disorder as a whole. We learned a lot about what was really happening inside Bertrand as a consequence of those tears. And then it sort of emboldened me to keep going, say, well, what else can we do if we can do that? And now things started to get a little more computational with time. Because what I did there was a lot of old-fashioned biology and biological science to come up with that prediction. But I felt like there was more that we could do with computation and more data. So I actually somehow picked up a grant to do full drug development for this disease. And what we did is we built a model for the disorder using planarian worms, meaning we genetically modified planarian worms to have the same disease as Bertrand. And using these, we actually identified a druggable target within their genome. It's another enzyme, it's called NGase, and what we ended up needing to do was find a way to inhibit NGase. So we needed to find a drug that would act as an NGase inhibitor. Now, to do that became entirely computational. We took a 3D structure of this target. We identified where the charges were, and we were basically looking for some small molecule that was inverse in shape and charge to this pocket on this protein. Basically inverse in shape and charge to the catalytic domain, because that meant maybe it will actually serve as an inhibitor for this molecule. So this was 100% computational at this point. So we tested 200,000 different structures in these simulations. And in doing this, we identified 70 compounds that looked like they were potential inhibitors of this target. So from there, we actually noticed that 14 of them were already FDA approved. So 14 were compounds that we technically could give to Bertrand the next day. So what we did with those is we took them to the bench and we used mass spectrometry to see if any was a true inhibitor of this target, and one in fact was, and that happened to be Prevacid. So Prevacid is an acid reflux medication. I looked it up. It's called Refluxon over here in Hungary. You can buy this in the grocery store in the US, and so that's what we did for Bertrand. So while all that was happening, other stuff was happening that led ultimately to what I would say is now this algorithm for doing precision medicine at scale. So I ended up starting a company that was acquired last year that focused very specifically on doing drug screens for ultra-rare genetic epilepsies, specifically those with a defect in an ion channel. And then I got the offer of a lifetime. So I got called to the White House. I met with President Obama. He said he'd read the article in the New Yorker and that he was wondering if I'd be willing to help out on the launch of this new precision medicine initiative that he had in mind. I kind of stood there for a couple seconds and said, yes, I'd be more than happy to do that, President Obama. I will do anything you want. If you want me to stand on my head in a corner, I will do that too. Just tell me. So I did that, ended up drafting some of the founding white papers around the precision medicine initiative before it's launched in the US. And then it launched. And what the Precision Medicine Initiative is doing is it's collecting the health records of 1 million Americans along with their genetic data so that we can build what I would call the Rosetta Stone of the human genome. Because right now, I can sequence any person's genome in this room, and we're going to find around 20 million mutations that make you different from somebody else in this room. And what I can't do for almost all of those mutations is tell you what, if anything, they actually mean in terms of your health. But if we collect enough people, then we can use this as a lens to say, well, actually, when you have mutations in this gene or mutations that look like this is what they do in aggregate to human health. So that's the purpose of this initiative. I worked a lot on the sister initiative that was part of this called the Million Veterans Program. The reason I did this is they already had 500,000 veterans enrolled in it. I ended up launching a micro initiative inside of the Precision Medicine Initiative, designed really to do a proof of concept that you could do what I'd done for Bertrand all over again. We called it PETMA, or the Patient Empowered Precision Medicine Alliance. And the goal here was, again, to quickly identify treatments as rapidly as we had, or even, in fact, more rapidly than we had for Bertrand, but do it for other diseases. So I'll give you a computational example of the kind of things we did under this initiative. So there's a disease, it's ultra rare. We actually helped the dad discover this disease in the process. It's haploinsufficiency, meaning you have half the amount that you need for this gene, USP7. And a company called Recursion Pharmaceuticals volunteered in this case to do a drug screen on this. And it's a very computational process. What they did is first they use computer vision to build a morphological fingerprint, if you will, of a healthy cell. Then they give genetic disorders to disease cells, and they re-image them, and they look to see what features have changed morphologically. Then you can just try lots of different drugs to see if anything out there actually makes a difference in terms of a given cell, or in terms of a given disease. So this was done for this USP7 disorder, and in fact, they identified these heat shock protein inhibitors that looked like they rescued the structure of the cell, and is now potentially on its way to actually helping that patient. More recently, actually, using an AI I'll talk about in just a moment, we also identified other treatments that could upregulate the working copy of USP7, including a relatively common drug like metformin. So as the Obama administration drew to a close, I was able to go back with several examples and say, it looks like this vision of precision medicine where we tailor treatments down to the molecular level using all the high-resolution data we can get on a patient. It looks like it's real. So then, you may recall, there was an election. And oddly enough, I actually got an invitation to spend another year around the White House. So I did that. Actually did spend a year in the Trump administration. And after a year of that, I think I'd had enough, done about all I could possibly do, so I went ahead and quit. And oddly enough, 2 days later, the US government shut down. I don't know if there was a connection or not. And people do ask me, what was it like working for the Trump administration for that year? And I think all I can say on the record is that, well, on a day-to-day basis, it was interesting. And I gather from news reports that it remains interesting to this day. So if you want to know more, I do have a policy of 1 beer for 1 story conversion ratio, and I'll be around later tonight. So then I got another amazing offer. I was offered the directorship really to create this institute from scratch, to build this Precision Medicine Institute down at UAB. That's in Alabama. And they said, we want to see if you can scale up your vision for precision medicine. Basically do for everybody, for every patient, whether it's rare, cancer, common, chronic, you name it, what you've done for him. So that's where I've been doing that now for 2 years. So how do we do this? How do we actually find that ultra-individualized treatment for everybody? Well, a lot of it is actually drug repurposing. We have to find some drug out there that maybe is meant for some other condition and reuse it for a novel condition or in the context of an individual patient because it's just what works for them. So one way to do this would be to do what are called drug screens, where you actually try to find biologically, chemically speaking, a drug that works for a given patient. So the way this works is you take a patient, from them you build a model of their disease, and it could literally come from them. It could come from their blood, it could come from their skin, or it could come from their data. If you have a genetic report on them, you can build a genetic model in many cases of their disease. And it could be a cellular model, it could be a model organism like a fruit fly or a zebrafish, those all work too. But the goal here is to identify, using some experimental apparatus, some kind of signature of the disease in that model. So you want to observe what we call a phenotype. Then you can combine that model of the disease with different drugs. In fact, you can combine it with all approved drugs at that point and check to see if anything rescues the signature of that disease. So that's what we do. For an example of this, I won't go into this because this is very wet biological. We did this for SCNA epilepsy, these ultra-rare epilepsies, where we actually took kidney cells and we put the mutation from a patient into the kidney cells and then observed the electrophysiological disturbances in conjunction with all approved drugs to see which approved drugs out there would work for those patients. Ended up significantly improving, or if not, or in many cases actually eliminating their seizures, and you can read about how we did that if you want more details. Just to show you something where the wet and the computational have really intersected, I'll talk now about a kind of experimental apparatus that a company called Q-State Biosciences can do. Full disclosure here is Q-State is the company that bought my company. But what they have is probably the most high-resolution model of neurological disease that's ever been created. And it generates ridiculous amounts of data. In fact, just managing all the data coming out of these reads has become one of the biggest engineering challenges at the company. More so even in many cases than the amazing biological stuff they had to do even to get to this point. So what they can do, they can take your skin, they can turn that skin into a stem cell, they can turn that stem cell into a neuron, and in the process they can also insert 2 additional genes into that cell line. One gets excited by blue light. So the neuron will fire when you hit it with blue light. The other one will emit red light once it's excited. So what you can do, once you've done this, is actually individually excite neurons and watch as signal propagates between them. And so this is what you're watching right here. You're watching a purposefully excited neuron and then signal propagating out from that. So it turns out you get signatures of a lot of neurological diseases this way. And so once you have that, you can screen drugs against them to see if anything actually corrects it. And it to me just blows me away that this is possible at all. The fact that we can have this level of resolution on a neurological disease. It would have been science fiction 5 years ago, and yet here it's something now that we're doing almost routinely. The other way to do it, I think, which is more interesting here, is to use artificial intelligence. And this is what I've invested most of my energy in terms of doing this at UAB at scale. So to do this, one of my faculty, Will Bird, has built a system called Medicanron. And so Medicanron is really all about fusing logical reasoning with biomedical knowledge. And under the hood, there's a relational programming language called Medicanron upon which all of this is built. So if you've never heard of Medicanron, think of it as Prolog on steroids. That's really what it is. So really what we're trying to build here is to take old school logic, to combine it with high-speed automated reasoning to achieve superhuman-like deduction with clinical insight. That is the goal of Medicanron. And what you'll notice is that when I'm saying the word artificial intelligence now, I'm actually using it in the 1980s sense of the word. So I'm talking about sort of the purely logical view of artificial intelligence, where you use logic to collect knowledge and infer new facts. So you can ask it very low-level questions. You can say, hey, what's an inhibitor for X? Or you can ask it very high-level questions, like what may treat this disease X? And for example, if you ask it for treatments for overactive bladder, it can generate 145 possible treatments for overactive bladder. And if you ask it for treatments for diseases that don't have treatments yet, it can still try to guess what treatments there might be. So for example, for Fanconi's anemia, it suggested xylitol, which is a sugar alcohol found in things like chewing gum. And the reasoning it generated here was that xylitol will inhibit alcohol dehydrogenase. Alcohol dehydrogenase creates the reactive aldehydes that cause the double-stranded breaks in DNA that Fanconi's anemia patients can't repair. I say all that because there's a clear logical argument beginning to end for why it makes this prediction, which stands in contrast in some cases to the explainability of more modern AI techniques. But if you're going to actually give a report to a doctor and say, I think you want to modify the treatment of this patient, you really need to have a tight logical argument behind why they should do that. So that's why we like this approach. So to do this, we just used a lot of natural language processing. It actually pulled in 30 million abstracts from PubMed and processed all of them into knowledge triples. So that inside of it, what it really has is this knowledge graph that has relationships between things like genes and drugs and symptoms and diseases, and then, again, relationships between them. So for example, you will find in this graph, Prevacid inhibits NGAze. That's from one of the papers that we published. And there's lots of other edges in there now, too. But there's also inferred edges. So, for example, if it knows that blue increases red and red decreases green, it can infer that blue decreases green. So if we're trying to inhibit a target like green, but we can't find a direct inhibitor, we can always back up and look for other ways to inhibit that target. And we actually do this quite frequently now. So you build these knowledge graphs, and you can mine them for logically significant, which are then biologically significant relationships between concepts. So to actually get this out there to patients and actually deploy it for them today, we've embodied this in terms of what we call a research consultation service, that patients can literally, from all over the world, they can just email us and say, hey, I don't know what to do in this situation, what would you do? And we try to identify the next step for them on their therapeutic odyssey, if you will. So the goal is to always be able to find the next step for any given patient. And to do this, we follow a step-by-step process, and this is, in fact, the algorithm for precision medicine. So I'll show it to you now in high-speed flowchart form, and then I'll talk about the inner guts of this algorithm and where we spend most of our time on it. So it really begins with precision diagnostics. We try to go from often genomic data into that root molecular cause of disease. And once we're inside, and once we actually have all that data, if we find the cause, if we find the gene driving the bad behavior, then we'll ask a question of it. We'll say, okay, as a consequence of this mutation, is this mechanism, is it now overactive? Is it underactive? Is it absent? Or is it toxic? So this is really the heart of the algorithm for precision medicine. Once we've figured out how a given mechanism has gone haywire, then we have a bunch of strategies behind each of these for finding the appropriate thing to do. For example, if a mechanism has gone overactive, then we need to inhibit that mechanism. If it's underactive, we need to activate the mechanism. If it's absent, then we need to compensate for the absence of that mechanism. And if it's toxic, then we need to find a way to eliminate that mechanism. So this is the essence of the algorithm for precision medicine right here. So we've been able to scale this up to the point where we started this about a year and a half ago. We've now seen hundreds of patients in the last 12 months and engaged them in this process of identifying their next step. And in terms of scaling this up, it's actually been quite useful to use students. So I actually use lots of undergraduates as part of this process to interface with the tools, which is surprising because you think if we're computer scientists, should we try to automate everything? And we will. We're slowly automating everything. But one of the reasons I focused on undergraduates is, first of all, they're everywhere. They're ubiquitous. And secondly, they're incredibly cheap. So you can reliably find them anywhere you go. So this is the team we have right now. In fact, they're not all undergraduates anymore. In fact, the one on the lower right is actually an MD PhD student. The one on the upper left is now my PhD student as well. But they all started off with no more than an undergraduate background when they first started working with me. And they do a variety of things. So they'll manage interactions between the physician and the patient and the AI tools, and then the scientists that have to do experiments on the patient's behalf as well. So they're very engaged as case managers on each case, and they do a lot more than just run queries in an AI tool. And just to give you an example of some of the other stuff we do to optimize the process, my student Jordan has actually invested a fair amount of time in a pretty customized Airtable implementation so that we have a really efficient process for doing case reviews, so that as we go through the hundreds of active cases each week, mostly we're just saying, no update, next. And we can figure out for any given case, if there's any new action that needs to be considered in light of any additional data that's been collected in the prior week. We also have a lot of great faculty and staff, and they provide a lot of individual expertise into this process on behalf of individual patients. And now I'll just give you some examples of the kinds of things that we can do with this group and with the technology we have. So May was working on a case for a patient that had a variant in a gene called TMLHE. You can think of a variant as a mutation, and reasoned that the disease is actually being driven by a loss of function in this gene in TMLHE. Which resulted in turn in a carnitine deficiency, because this is in the carnitine biosynthesis pathway, which in turn meant that this patient needed to go on supplemental carnitine. So this is an example of the kind of reasoning that these case managers will do in conjunction with the tools that we have available. Jillian had a patient with a mutation in a gene called ROBTB2. She reasoned that because, in fact, this mutation was in a region that controlled the degradation of the protein, that the protein was insufficiently degraded, resulting instead in an upregulation of the amount of this gene that was present. She couldn't find a direct downregulator of the gene ROBTB2, but she found the regulator of ROBTB2 and found that that could be downregulated using a drug called celecoxib. We had another patient with what's called a variant of uncertain significance, and a gene called MAPK8IP3. Lindsay realized this was actually causing a haploinsufficiency in MAPK8IP3. And in fact, when she used Medicamerin, she found that vitamin A would be a potent upregulator for this particular gene. So if you take it at nearly toxic but not toxic levels, it looked like it was beneficial in this case. And actually, in that particular case, seemed to have been extremely beneficial, because that patient went from non-ambulatory, non-walking, couldn't even stand, to within 6 months being able to walk and stand at about 5 months old. And then another case, I think, that highlights sort of the flexibility of the tools we built was actually one that Jordan had on intractable cyclic vomiting. So in this case, it was a 19-year-old woman. And the other reason I like this case is that this is not a genetic disease. This is a disease, there's probably a genetic basis for it, but we didn't have a clear genetic mechanism to go after, and it forced us to use our tools more creatively. So she was 19 years old, and she had multiple episodes of vomiting per day. In fact, since age 11. She weighed 78 pounds, which is extremely underweight because she was about 5'4", and she had tried effectively all known treatments. So at this point, her parents reached out and said, we've tried everything. We don't know what to do. But she can't keep food down. She's always in the hospital. We're very worried about her. So it turns out that when somebody says they've tried all known treatments, especially for something as common as vomiting, it's almost certainly not true because there's a lot out there that is known. There's 29.1 million published papers. 64,000 of them are on nausea. 4,000 of them are on treating nausea. Between them, there's 374 distinct treatments that you can try. So no one has ever tried everything for nausea, I guarantee you. So if you look at the top end of the histogram, in terms of treatments by the number of publications that support them, you'll find 169 papers for Zofran, which is the most popular anti-vomiting drug that there is. But if you keep looking down this histogram, you will find regions that people don't, even experts don't know about. We call this the unknown known. So if you keep going down the curve, you eventually end up in a region where you find nasally inhaled isopropyl alcohol. So this is the kind of rubbing alcohol that people keep in their bathroom cabinets that you put on cuts and scrapes. And we found 3 papers that said that if you sniff this while nauseated, it could alleviate the nausea. And so we turned this over to her team, and they recommended it, and sure enough, it actually worked. So after years and years and years of intractable vomiting, came across this unusual remedy, supported in the literature by 3 papers, that worked for her. And she at the time that she last reported in, gone 8 months without vomiting, still hasn't vomited. It's been about a year now, and she's up to 125 pounds. But not only that, because she's out of the hospital, because she can control these episodes of nausea, she was actually able last April to finally get married. So she married her high school sweetheart and moved to California. So if you're interested and you happen to be in Alabama, you are more than welcome to come join us. We meet every Monday to do case reviews as a team. You'll see us deploy our tools in real time. And in fact, we've been so successful that we're moving into the UAB health system, so we won't be just a research project. We'll actually be a service within the health system at UAB. You may be wondering if this tool has made predictions for Bertrand, and it absolutely has. So if you ask Medi Canron what we should do for Bertrand, it reasons that in order to compensate for the absence of NRF1, which is activated by Angli1, you should supplement or try to upregulate NRF2. And to upregulate NRF2, what you really need is broccoli. Or more specifically, a component within broccoli called sulforaphane. So he now takes these condensed sulforaphane pills to help upregulate NRF2. And actually, this seems altogether in aggregate to be making quite a difference for him. So he's a happy kid day to day. He still has his struggles. It's still a very serious disease. But he's definitely lived far longer than all of his original life expectancies that were handed out. And he's actually making some forward progress in some dimensions. So he's getting better and better at communicating. He's actually finally, in the past year and a half, really started to master this eye gaze computer and to talk to us really for the first time. So he actually asked for a pet fish a couple of years ago. And, specifically, he asked for a whale shark. So it is a little bit outside my budget, but we did manage to get him an aquarium for his room. So, but I want to make the case here that I've talked a lot about rare diseases, but this goes well beyond rare. It impacts things like cancer, too. So we have a precision oncology program where what we do here is sort of a front line is we pull out tumors, we sequence the DNA inside the tumors, and we look for the mutations that are driving an individual tumor. And then we try to match those mutations to medications that will cut against the effect of those mutations, which, when it works, can actually be remarkably powerful. So I'll share one story here. We had a patient with prostate cancer who'd undergone 42 rounds of radiation and 17 rounds of chemo with no effect on his prostate cancer. So he's basically at death's door, gets enrolled in our clinical trial. We sequence his tumor, and we find that genetically speaking, while he did have prostate cancer, it looked a lot more like the genetics of ovarian cancer, despite the fact that he's a man. The genes that were overactive or the genes that were mutated look a lot more like what we see in ovarian cancer. So he actually went on an ovarian cancer drug to treat prostate cancer. And it worked. So it's been over a year now, and he's effectively cancer-free. So for patients that go treatment resistant, we have other strategies. So Chris Klug is an investigator at UAB, and he does what I would call essentially a brute force attack against cancer. So if you've got leukemia, he will take your blood. He will then start testing your blood against different approved drugs and drugs in clinical trials to see if any of them will cure your leukemia. And so far, he always finds something that works in the context of any given patient, even when they've gone treatment resistant. So I'll close with one last example that pulls together absolutely everything in the context of precision medicine. So it's actually back to Bertrand again, and this is from last May, so it's very recent. I took him to the Science Museum on Star Wars Day, took him down to the aquarium, and by dinner time that night, he was near dead from septic shock. Again, this is back in May. So what began, I didn't realize it at the time, was a second diagnostic odyssey. So while he's being admitted to the ER, and they're saying this looks like septic shock, I'm asking questions, because I'm not a physician. I just have to use this tool that we built to figure out what this actually means. So I say, well, what causes septic shock? What are the consequences of septic shock? And ultimately, what are the treatments for septic shock? And right away, something popped up and caught my eye when I looked into that region of the unknown known. I saw albumin. And the reason this stood out to me is that Bertrand, according to the studies we've done, in fact, all the patients with this disease have chronically low albumin. And if you look in the literature, if you have septic shock, low albumin is strongly correlated with dying. So you really need to get Bertrand albumin right away. Luckily, the physicians believed me. I could say, here's a paper and here's a paper. If you put these two papers together, he needs to get albumin immediately. And he got it. So he survived that very first night. I mean, and septic shock, even in the best of conditions, has about a 50% odds of survival. And in his case, it was probably worse than that, given that he was low in albumin. But he actually made it through that first night. And that began the first week in the ICU. Unfortunately, he got antibiotics before they took his cultures, or before they took the cultures for his blood for the culture. So none of the cultures were coming back with any organisms. We didn't know what it was that was driving the disease. But by the end of the week, he was stabilized enough with oxygen support that he was trending in the right direction. And after the end of week 2 in the ICU, he was showing slow improvement on, again, lots of antibiotics and supplemental support. And he looked like he was almost good enough to go home. So they actually did send him home on oxygen and a slight fever, but he was back in 24 hours. He had extreme pain. He ended up in the ER, and that began week 3 in the ICU. And now things kind of went off a cliff, because now he had a bone infection. He had significant swelling in his abdomen. He had pleural effusion, meaning fluid outside the lung that ended up collapsing his lung. And the cultures were still negative. Again, he'd been on so much antibiotics at that point that it was going to be difficult to figure out what it was. So he had 2 surgeries to clean out the bone. He needed to get a chest tube. And the day he was supposed to get the chest tube, he actually crashed. So this is him in the morning. And then 4 hours later, he coded in the ICU in respiratory distress. And they just barely managed to bring him back, and to the point where it was so bad that my wife and I thought, this is it. This is probably the last we'll see of Bertrand. So we actually just effectively said goodbye to him that night, not even thinking he would survive. So this is his mom basically saying goodbye. And I just begged him all night long to operate, to get the chest tube in. And when they finally came time to operate, his hematocrit was actually too low. So to even get to the operation, to get the chest tube in, to drain the fluid off his lungs, he needed a blood transfusion, which he got, which he survived. And he did actually get a chest tube, but it was too small. So he coded again in front of the physicians in the ICU, and they put in a bigger one. So he made it through. Unfortunately, we're now week 3. The cultures are still negative. I mean, we had no idea what this thing was that was killing him. So it turned into a holiday weekend in the US, and the doctor came into the room and said, well, I'm sorry, but nothing happens in hospitals on weekends, and especially nothing happens in hospitals on holiday weekends, so we're just going to keep him alive until Tuesday. So at that point, I said, look, it's time for me to step in and do something. I preach precision medicine. I need to practice it here. So I said, it's time to write some code. So I created a diagnostic extension to Medicanum. And the way this worked was it looked for relationships between diseases and symptoms and said, give me a set of symptoms. I will rank the diseases in terms of most explanatory power that could cause those symptoms. So if you can explain all the symptoms with one disease, that's up at the top. I came up with this list right here. And then we realized that we had to start ruling these out quickly to figure out which one it actually was. So we started aggressive testing for these, but at the same time also used a really brand new technology called metagenomics. So your metagenome is your genome plus all the other genomes of all the stuff floating around inside of you. Because all of us have lots of stuff floating around inside of us at any given point in time. So if we pull that out and we look at the DNA content of what's inside, we get a glimpse of what's inside of you. And again, using DNA as a proxy for what is actually there. So the question was, can we find the bug using metagenomics? So literally checked his blood and pleural fluid out of the hospital and relayed it up to a genomics facility in Huntsville, Alabama, via my associate director at the Institute, Andy Krause, who got it to Sean Levy, the director for genomics at Hudson Alpha, who actually did this sequencing on these fluids. We took, again, blood and pleural fluid. We did 40x shotgun sequencing 4 times over in 3 days. To put that in perspective, Bertrand's original diagnostic sequencing took 2 years on 2% of his genome. Here we're doing 100% of his genome 4 times over in 3 days. So that's how fast things have changed in 9 years. So what was it in the end? Well, it's a lot of stuff. Again, we're all infected with a lot all the time, most of it not harmful. So if you looked at the phylogenetic distribution of what was inside of Bertrand, this is what you get. So there's a lot of stuff inside Bertrand at this point. The question is, which chunk in this pie chart was actually the one driving the infection? Well, if you intersected that with the results of Medicanron, only one thing remained, and that was pseudomonas, which is something that you don't typically see in healthy people. You do sometimes see in immunocompromised people like Bertrand. And then the question was, can we prove that this is really it? So the idea was to localize a pathogen using some novel experiments. And the idea here was to burst all the cells in both samples to see if we could figure out where this thing was and really what it was. So again, if you're using DNA as a proxy for what's there, you only see it if you actually have DNA for it. So for example, this green organism here, because it hasn't shed its DNA, you wouldn't see it if you sequence that tissue. But if you actually take that sample and you burst the cells and then you sequence, now you'll see it. So that was the idea, was to look for a shift in the distribution of organisms before and after bursting all these cells to see what was actually there in that tissue or in that sample itself. So when you did this to blood, nothing shifted, but when you did this to the fluid from his lungs, it did. We saw a significant increase in the share of Pseudomonas once again. So the predictions were matched by what we saw in reality. So it was almost certainly Pseudomonas. The other thing we saw, if you look at the data, was that there's a virus that eats Pseudomonas floating around inside of Bertrand too. So if there's a virus that eats Pseudomonas, there's probably a lot of Pseudomonas to eat. There was also an above average amount of E. coli present, and so the solution here became a rarely prescribed antibiotic called meropenem that would kill both, depending on which one it was, and that actually worked. So after 4 weeks in the ICU, 48 hours later, he was out. The pleural effusions were receding, the ascites were diminishing, his needs for oxygen support were dropping, and in fact gone pretty quickly. And when we did another round of metagenomics, neither Pseudomonas nor E. coli was present. So this is him learning he's about to get out of the hospital. This is him getting home, getting reunited with his fish, which he was very, very happy to see. And 1 week later, actually took him down to the Gulf Coast in Florida and took him swimming with the dolphins, which is something that I'd promised him that we would do once he got out. So in the meantime, we actually got a clinical trial funded by NIH to do an experimental therapy that'll be starting in February for his disorder. And my closing thought here is if any of this inspired you, if you yourself are interested into moving from data science, computer science into medicine, you can. And now is absolutely the time to do it. There is a desperate need for computer scientists, not just computer science, in medicine. The one book I always recommend to folks is Quick Start Molecular Biology. It's really written with scientists and engineers and computer scientists in mind. So that, basically everybody in this room, I think, is a good candidate for it. And there's lots of immediate needs where we could do so much better with a little bit of computer science or really computer scientist. For example, we need to do much better NLP of the literature than we currently do. So I'll close with that. I will attend the office hours later if you want to chat some more, and I'll also be there at the party tonight. But with that, I thank you and welcome any questions you may have. Thank you.
I think I'll read this out to you. You are a good dad. I'm going to personally amend that. You are an amazing dad. What a phenomenal story.
I'm not sure my other 2 kids would agree.
So how do you fund your work on seldom cases, as those won't bring a lot of ROI based on patient numbers?
To fund the work, it's a combination of philanthropy and NIH funding. So we really do treat every case essentially as a science project. And so we can argue that there's scientific merit to helping these patients out. And that's been the angle we've taken so far. And again, we've been very successful in getting NIH funding for that.
We're a little short on time, so we'll just do this next question.
Okay.
Or maybe we're not short on time. Could personalized medicine be used to optimize the functioning of healthy individuals in order to achieve superhuman powers?
Yes. But it's complicated, obviously. So I mean, this kind of stuff it's in the early stages. It's obviously highly controversial. It's by no means easy. So right now, if you wanted to edit the genome of a human being, good luck with that. Gene editing technologies like CRISPR, you can do it in a single cell. But if you even could deliver CRISPR and edit the genome of every cell in your body, given the error rates in CRISPR, do you know what you would have instead of superpowers? Cancer. That's what you'd have. So it's not recommended, but it's conceivable that we could identify, targets within the genome that you'd want to give to confer advantages and things like that. But that's well beyond my pay grade. And the ethics involved in that are obviously, tricky, to say the least.
Okay. Would precision medicine with viruses such as HIV, herpes, etc., something?
Would it work? Yeah. So, I had a postdoc come to my office not that long ago, and what she was doing is she was actually analyzing the genomes of HIV, and she had thousands of HIV genomes to analyze, and she was looking for patterns in the glycosylation on the surface of HIV, which is how HIV, and you can think of glycosylation as a way to defend against antibodies and things like that. And HIV has shown its ability to rapidly mutate this glycan shield on its surface in response to any kind of attack from an antibody. It's why it's so hard to kill. And she was looking for patterns in how if you target this glycan, where does it move? What happens in terms of the reshifting of this glycan shield? And what she found is that it's not random, that there actually are constraints on how these glycans can reconfigure themselves in response to selective pressure. But the way she found that was she came to us and said, right now the way I'm analyzing these genomes is I paste them into Microsoft Word, and I use search and replace to do my genomic analysis. And we thought, oh my God. So we wrote a Python script that afternoon and finished 3 years of her PhD, literally in an afternoon. So when I say biology needs more computer scientists, that's what I'm talking about.
Wow.
Your Python script could do a PhD in an afternoon in some cases. It literally can, because you know how to do it. Whereas these poor biologists are using literally Microsoft Word and search and replace to try to figure out how to analyze a genome. Yep.
Wow. When will precision medicine be widely available and covered by public health insurance in the European health system? I'm not sure. Offer information there.
I have no idea. But I do think that the place we're pressing right now to get this covered is the payers, to say that there's an advantage to payers in doing precision medicine, that in many cases we can find better solutions that are less costly than standard of care. So payers have a lot to benefit in addition to patients from this.
Okay. Any good news on Alzheimer's that you're personally aware of?
So I do have one project on Alzheimer's funded by the Alzheimer's Drug Discovery Foundation. Here what we've done is we've taken an ultra-rare driver for Alzheimer's. It's a gene called TREM2. So if you have certain mutations in TREM2, it tends to generate really aggressive early onset Alzheimer's. But it also looks like modulation of TREM2 would be beneficial not only for the patients that have TREM2-driven Alzheimer's, it looks like it would be broadly beneficial for everybody that has Alzheimer's. So by focusing on these rare cases of Alzheimer's, it looks like we've identified a druggable pathway that could help every patient with Alzheimer's. So what we're trying to do is do exactly that. We're looking, using the computational tools I just showed you, the way we found Prevacid, to look for modulators for TREM2. If we find that, there's a chance, again, that everybody who has Alzheimer's could benefit from that drug, not just the TREM2-specific patients.
Wow, okay, and I think we've got time for one more question. Would this methodology work on brain diseases such as tinnitus, or is this more applied to physical diseases?
Yeah. Well, tinnitus being caused by actual physical trauma to the ear, not likely, unless we can replicate a model of that trauma and do drug screens against it. I think it's generally better, at present at least, used best for diseases that have a clear molecular basis. That said, if somebody approached me with tinnitus, what I would do is I would ask it to generate the histogram of all possible treatments for tinnitus that have ever been found in the literature, and make sure that if nothing had worked for them, that there wasn't something that they had missed. So sometimes just a better consideration of what's already out there goes a long way for patients. And I have the tools sitting on my laptop right now, so if somebody does have tinnitus, please come up and see me and we can run that query.
Okay, and on that note of the invitation, you can find Matt in the Prezi booth, which is just behind the information booth, for office hours for the next 20 minutes, and at the party tonight. Thank you so much,
Great.
Matt. What a
Thank
wonderful
you.
story.
Appreciate it. Thanks.